I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 184th episode of this podcast dedicated to the care and feeding of your very own brain. In this episode, we're going to be getting back to our roots, talking about a cognitive compound, something called Theacrine, not unrelated to a compound that I'm sure you've heard of called caffeine. You might hear me pronouncing it differently across the course of the episode. When I look at the word, it kind of reminds me of creatine, so I probably say it a couple times that way, but I believe Theacrine is actually accurate. In this matter and others, I will defer to our expert on Theacrine, Dr. Richard Bloomer, who I'm going to be speaking with in the main interview. If you hang around until the very end of the episode in the Ruthless Listener Retention gimmick, on the off chance that you only listen to podcasts, I'm going to tell you about a fascinating study showing what learning to read late in life does to the brain. It's never too late to learn to read. It's never too early to move on out of the intro segment. And I will do that now, heading on as usual to This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So you may or may not have heard of the term binocular rivalry, but this is a term that kind of makes me think of the old comic Spy versus Spy and the two spies pulling out binoculars and trying to coordinate doing something dastardly to the other one. As you might imagine, that's not actually what binocular rivalry is, at least in the scientific sense. Binocular rivalry is what it's called when your two eyes are shown something different, and then your brain is left trying to figure out what to do, and there's a couple of ways of handling that problem. One is to sort of switch back and forth between the images, make the eyes take turns. First, we'll show what's in the left eye to conscious awareness, then we'll show what's in the right eye, just sort of switch back and forth. That's what normally happens for most people. The other way that the brain might handle this is to merge the two images and basically give you sort of an overlapping combined version of what your left and your right eye are seeing. The researchers in the study that I'm about to tell you about, they did this binocular rivalry experiment where they showed a red patch in the visual field of one eye, a green patch in the visual field of the other eye. So study participants could alternate between a red and green flashing Christmas light sort of thing, or the brain could split the difference, combine the two, and give them a muddy brown patch. As we all know from a color wheel, red and green make brown. Researchers at the University of Melbourne in Australia gave this test to 123 volunteers, along with tests in the Big Five personality traits, trying to identify where each person fell on a personality spectrum. Personality is obviously a tough thing to quantify, but most psychologists these days break down the Big Five personality traits as extroversion, agreeableness, conscientiousness, neuroticism, and openness. And for a lot of reasons, it seems like openness is the most interesting one, at least in the world of modern psychological research, because it seems to be subject to change. We talked on a much earlier episode, I don't remember which one, that the use of psilocybin, the psychoactive ingredient in magic mushrooms, tends to increase a person's score on openness, and they see that change in personality maintained years after the experiment took place. So kind of interesting. But in this recent study at the University of Melbourne, what the researchers found was that people that scored higher on the personality trait of openness were also much more likely to see the brown patch rather than alternating red and green patches in their visual field. Openness, by the way, the definition of that personality trait in this context involves creativity, imagination, and being willing to try new things. So a difference in personality corresponded to a low-level perceptual difference of physical reality, which is pretty interesting if you think about it. This is maybe reminiscent of that thing with the dress from a year or so ago. Some people would perceive it as a blue and gray, and other people see it as white and gold. It'd be interesting to run that study, see if the perceptions of that dress vary with personality type. But as far as this binocular rivalry, a higher score in openness corresponds to a greater likelihood that you will use the merged field rather than the alternating distinct field solution to the binocular rivalry problem. The study's lead author, Anna Antonori, said, "The mud-brown version was a somewhat more creative solution to the problem presented by the two incompatible stimuli. The gate that lets the information reach consciousness may have a different level of flexibility. Open people appear to have a more flexible gate and let through more information than the average person." I'm not 100% sure that I agree that alternating red and green flashing is not an equally creative solution to the problem, but that's just my opinion. And full disclosure, I'm one of the people that missed the gorilla walking through the video in that black and white bouncing ball experiment that you've probably seen on YouTube. So this study is the first research showing differences in personality being tied to direct low-level perception of the world, but it's not exactly clear why this is happening or what's going on. Antonori speculates the chemicals within the brain may have overlapping roles in both perception and personality, but as of now, nobody really knows.
Smart Drug Smarts, the podcast so smart, we have smart in our title. Twice.
picked up a couple of 5-star reviews on iTunes. Listener Giant Meat Hog from the USA said, I'm a mailman who listens to podcasts all day. Smart Drug Smarts is by far the best podcast for dialing in my cognitive function. I've learned so much from the show and the guests who are interviewed. And listener Aoli 2000 from Sweden says, found this podcast through my interest in nootropics, but it offers so much more than that, covering a lot about our brains. Well, thank you, Aoli and Meat Hog, and everybody else that's been leaving reviews, all very much appreciated. I've also gotten a number of emails this week from people that are taking me up on the offer, or at least dipping their toe in the water to see what's there next. I mentioned last week that Water Fast Week 2017 is fast approaching, just about a month now. Going to be kicking that off on July the 8th, going to the 14th for people that make it the full 7 days. We'll be talking about that more in the coming weeks, but if you want to sign up for more information, you can do that online at smartdrugsmarts.com/waterfast. Waterfast is all one word. And I can absolutely guarantee you an interesting time, maybe not a good time, and a lot of stomach rumbles, but not a time you will soon forget. For those of you that just want to sign up for something but have no interest in not eating for a week, you could always sign up for the Brain Breakfast newsletter. That is a standing offer. We've got our weekly or so newsletter of things near and dear to my heart and brain, things that don't quite make it into the show or are a little bit off in a weird direction, seeing where some of these topics touch in on neuroscience, and of course providing some links to neuroscience news around the web. We try to cover a lot on the show, but obviously not everything can make it into the episodes, and the Brain Breakfast can be a great place to catch that spillover. You can sign up for that at smartdrugsmarts.com/newsletter. Another thing that is not out of character for us to do on Brain Breakfast and our social media channels is to let you know about things we've got coming up over at axonlabs.io. Axon Labs is Smart Drug Smart's retail arm of things on the web. We've got a couple of supplement stacks over there, Nexus and Mitogen. Nexus built around aniracetam, CDP choline, and other compounds, a go-to for me on days like today when my work involves creative stuff. Aniracetam has been shown, among other things, to have significant boosts to short-term memory. And short-term memory I've talked about in other episodes, but it's worth coming back to. Actually feeds into creativity in an unintuitive but makes sense once you have it explained to you type of a way. Both Nexus and Mitogen can be found online at axonlabs.io.
Smart Drug Smarts.
So I'm about to be speaking with Dr. Richard Bloomer, who's the director of the School of Health Studies at the University of Memphis. He's also the director of the Cardiorespiratory and Metabolic Laboratory, and most appropriate to this conversation, the Center for Nutraceutical and Dietary Supplement Research. And we're going to be talking about theacrine, which is a small alkaloid molecule similar to, but obviously distinct from, caffeine, which occurs in at least some plants where caffeine is formed. It's not quite clear how common it is, mainly because people haven't been looking much for it, but it's worth learning about, and there's some interesting hints that it might have certain upsides that caffeine doesn't. This is one of these compounds that as you hear the conversation, you'll wish that there had been more research done to this point, but if there had been, theacrine would be a better known compound than it probably is. So consider yourself an early bird in even learning about this one. And with no further ado, let's jump in with Dr. Richard Bloomer.
Many of us working in the area of dietary supplement research and nutraceutical research are aware of one another and the work that's being conducted. We see each other at different meetings, etc. So I became aware of this through some reading that I had done, and then a colleague of mine who actually works within a dietary supplement and contract manufacturing business, he shared with me a product that he was looking into using the theacrine. So I became aware of first, I think, through that connection. And then from there started studying theacrine and really where it's been found in some of these different types of tea. I think this kucha, I'm not sure if that's the correct pronunciation, but it's Kucha tea. Unlike many of the other teas that are rich in caffeine, this particular tea seems to be very rich in the theacrine. I think at the dosage of approximately 50 or 75 milligrams per cup. So some individuals are probably getting it and potentially feeling quite good with the use of the tea and not even realizing what they're consuming. But that's how I first became aware of theacrine, and then we've been doing some work with it over the past few years, either as an isolated ingredient or in combination with caffeine, as well as within a kind of a mixed finished dietary supplement.
So biochemically, it's related to caffeine. When it occurs naturally, is it replacing the caffeine within the plant, or do they exist side by side to some degree?
You know, the different types of tea historically will have different combinations of the caffeine, in some cases the theacrine. So structurally, they are similar. I think theacrine technically is considered a methyl urate purine alkaloid versus caffeine, a methylxanthine purate alkaloid. They seem to have similar overall effects, albeit we find with the theacrine, people tend to not have quite the massive high and then the fallout a couple hours later, which is oftentimes what happens with a caffeine or the caffeine low. They tend to have much more of a stable energy and as well as a focus and just kind of that feeling of euphoria and focus, which I think a lot of people like, especially if they have to sit at a desk or do things in an office setting and they're using something like traditionally a caffeine. Maybe they don't want to sit down still after consuming a couple hundred milligrams of caffeine. They might feel like they need to get up and do things. So the theacrine does seem to help a little bit in that regard.
How long do you see for the period of effects? I know for caffeine, the half-life for most people is around 5 or 6 hours. Is it something similar for theacrine?
Yeah, that's what we're studying now. So that we have a paper, and I can't release a whole lot of details, but we have a paper that was submitted to the Journal of Caffeine Research, and we're finishing round 1 revision. So hopefully we'll send those revisions back in and everything will look good to the reviewers, and that paper should be published here in the next few months. But we essentially had 8 individuals in the lab and met with measurements over a 24-hour period. So we did a pharmacokinetic analysis with theacrine at 2 different dosages, relatively low and high, and then caffeine alone at a moderate dosage, and then the theacrine coupled with the caffeine. We tried to determine again in that study what the half-life would be, what the max concentration would be alone in isolation as well as in combination. And what we see is that in all individuals, it seems to behave in a pretty similar manner. And interestingly, the caffeine addition seems to prolong the overall effect of the theacrine. And I think that's of interest to a lot of individuals who tend to use the theacrine nowadays within these blended dietary supplements that might contain caffeine, sometimes a lot of other ingredients that maybe we're really not so certain as to how they're behaving. But just the simple combination of a relatively low dose caffeine, in our study we used 150 milligrams, so about the equivalent of a pretty good cup of coffee, coupled with the theacrine, seem to have a really favorable effect in these individuals in terms of how they feel, etc. And it did seem to prolong the overall time course of theacrine in terms of concentration. So I think that's of interest. And once that paper comes out, people will be able to see the details in terms of the half-life as well as the peak concentration at the different dosages.
Does the method of action within the brain seem to be similar on caffeine? I know it's supplanting adenosine and not letting your brain become aware that you've sort of got this mounting sleep pressure. Is theacrine doing something similar?
That's a great question. We haven't studied that. This whole area, at least from a human perspective, is what we'd probably say in its infancy in terms of study, because there's only a handful of published papers that have been done, and none of them have really looked at mechanism of action per se. I think our PK study that we just completed is probably one of the more sophisticated pieces of work, but certainly a whole lot more needs to be done in this area. Some of the animal work suggests specifically working on adenosine receptors and dopamine receptors, which may allow it for the theacrine to have that dual phase or dual component effect. But admittedly, we really haven't studied from a mechanistic point of view how exactly it's working, at least in humans at this point.
Is that something that's been studied in animals where there might be close parallels?
We haven't used it in animal models, but readers can look at PubMed and you'll see a number of papers that have focused on the theacrine in animals. And there are some really interesting findings. I mean, for me personally, because we do a lot of work with oxidative stress and antioxidant capacity, it has been shown to be a very potent antioxidant and anti-inflammatory agent. And I think when we look at human disease as a whole, it appears that oxidative stress and inflammation seem to be one of the main players for just about every disease we're aware of, from metabolic to cardiovascular to neurological. So I think that's interesting. Now, that said, the dosages oftentimes that are used in these animal trials are very much higher than we would see in a typical human subject, because we've been using dosages anywhere from 25 milligrams up to a couple of 100 milligrams. And I think there's a paper using 300 milligrams of the theacrine as well. But I actually just looked at a paper the other day, very interesting. It was in mice, and it showed that through oral administration of theacrine, it actually reduced liver damage that was induced through specific restrained stress on these animals. I thought that was a great finding, but when you look at the dosing, the dosage was, I believe, 10 to 30 milligrams per kilogram. They dosed over 1 week. I think it was a 7-day dose period. So one thing, whether it's theacrine specific or otherwise, I'd always caution listeners to focus on is when you look at these animal data, how relevant are they to humans based on the dose? Because if they're using 100 times what the effective dose would be in humans, you have to question how is that actually going to work if I'm using a dosage that's significantly different. That's very, very common in the dietary supplement world.
That phrase, the dose makes the medicine, the dose makes the poison, is always worth coming back to.
Right.
Is this a threat to the entrenched beverage industry, or are the ways that theacrine seems to avail itself in nature not something that you'd necessarily want to taste in their natural form so much? Like, are there equivalents to coffee or tea that somebody might actually want to drink?
A good question. As I mentioned, it's found in quantities that are pretty reasonable from at least the literature. A cup of this particular Chinese coca tea, this Kucha tea, contains roughly the amount of 50 to 75 milligrams per cup. So a couple cups of that tea would actually allow for a dosage that's pretty similar to what we've been delivering. Now, how that tastes, Jesse, I'm not sure, because I have heard from colleagues that embedding the theacrine into a beverage has been a challenge. Obviously, if people are going to consume something on a regular basis, it needs to taste pretty decent. And I haven't consumed it in a liquid form in the past, so I'm not certain about that. And I've been in the mentality that I'm willing to try different things, even if the flavor is not fantastic. But I think the average consumer, if it's not going to taste decent, time and time again, they're probably not going to consume it. It's much better to dump that stuff, if possible, into a capsule and get it into the gut that way.
One of the interesting, I'm not sure if this can be called a finding yet, but at least areas of research in theacrine sounds like it might not have the same tolerance that caffeine. You build up a tolerance pretty quickly. Can you talk a little about that?
Yeah, that's a great point. We haven't done any long-term treatment studies, but the group that did that work that you're referring to and looking at that paper, the authors did suggest that. And I think that is very interesting because, as you know, not just even with caffeine, but frankly with a number of different things. Years ago when people used ephedrine, you'd see the same thing. They'd start with a 20 or 25 milligram caplet, and quickly they're up to 3 or 4 of those caplets to get the same overall effect. And I know for a lot of people, primarily from a cardiovascular perspective, a blood pressure perspective, that was concerning. Now, again, there hasn't been multiple studies conducted to replicate these findings, but from the initial work, it does appear that it does not have the same sort of tolerance developed as you would see with something like a caffeine, where people need more and more to get the same overall effect. So I think that is an interesting overall finding, but we also need studies to replicate that and across different populations of individuals, from men to women to any potential user, old and young.
Have you seen any interesting or scary adverse reactions, things that people should be aware of?
It's another great question, because with this particular class of dietary supplements, if you lump it into the typical stimulant or class of supplements that are targeting focus, etc., generally that's been a concern. But we've personally done measurements, primarily of blood pressure with treatment. In this PK study, we did 24-hour assessment of these individuals, and heart rate. In other studies, we've done the same. We see a negligible increase, if any. In fact, some of these values have actually decreased across time. But from a blood pressure perspective, the most we're seeing is about a 2 to 3 millimeter of mercury bump with acute treatment, which is obviously small. And I know the studies that have actually done some blood measurements, metabolic panels and things like that, they haven't noted anything of significance either. So I think that's interesting because, again, so many individuals are using products or desiring products that could yield the sort of benefits that the theacrine has been shown to yield, but they are concerned about, oftentimes, does this elevate my blood pressure? Is it causing any sort of adverse cardiovascular effect? And at least to date, we haven't seen that. We haven't had any individuals that have any sort of adverse reaction to treatment.
One of the things that I've come across in talking with people about caffeine is every now and then it seems like there's a sliver of the population that almost has an inverted reaction to caffeine than what you would expect, where it makes them drowsy or sleepy as opposed to pepping them up. Have you seen anything paralleling that with Theacrine?
We haven't to date. That said, when we say we've measured individuals, we're talking maybe over the course of our studies, we've assessed 100 individuals. Usually, I think you'd want hundreds or thousands of individuals. And obviously, companies that are selling products containing these things, they may have additional data that they're collecting. But from a scientific controlled experiment perspective, we have relatively small sample, I think, of individuals. But that said, we haven't noticed individuals having that sort of reaction. Most individuals do tend to feel that their focus is enhanced, that their energy, concentration, overall mood is enhanced. But again, like anything else, it varies. Some people are claiming dramatic effects, and other individuals are claiming that, well, I might feel a little bit better, but I'm really not sure. So I think that's something that we see with all dietary supplements, some variability in response.
I was seeing in some of the studies, it sounded as if there was a bit of a dose-response inversion where small but measurable doses, it was actually calming for some people. But then after a certain threshold, it would start having the more caffeine-like stimulatory effects.
Yeah, I've read the same, and we in our lab haven't done that work. Now, we have used a couple of different dosages. In one of our studies, our recent studies, we used a low-end dose of 25 milligrams and then a high-end dose of 125 milligrams, so essentially 5 times the amount. But we didn't see any, as you're describing, major changes in terms of calming versus stimulatory. I know the other group has commented on that. They have used dosages, I think, they're upwards of 300 or 400 milligrams of the Theacrine. But our sample size, frankly, I think has been too small in our work to date, at least, to show that there's some differences in terms of the calming effect versus the stimulatory effects. In some individuals, certainly that's the case, but I think we always want to use caution if our sample's so small that statistically we're not able to really detect anything. But I think there's possibilities for that, and further work with larger sample sizes may actually show exactly what you're describing.
If somebody's curious and wants to try Theacrine in as pure a form as I guess they can get it on the market nowadays, where it's not necessarily mixed with too many different things, what is their best route for being able to take an N=1 experiment?
Well, again, that's something they probably have to do a little bit of digging on. I know there's a lot of products out there that are containing the Theacrine, but most of them contain it in conjunction with it. At the minimum, caffeine, in many cases, is going to be caffeine coupled with multiple other ingredients that really may or may not be having an impact. Purchasing the raw material and then actually dumping it into a beverage or something like that, that might be difficult if they're even able to obtain the raw material as an individual versus a research group or a university.
Yeah.
I think probably their best bet would be to search some of these online sources and see if they can get simply Theacrine encapsulated at dosages of 50, 100 milligrams, and then they can experiment with a relatively low dose to start and potentially move up.
Are there any people that you would recommend don't try Theacrine? Pregnant women or children under a certain age, people that should just probably stay clear of it?
I think probably we'd use the same cautious recommendation that we would have with most other things. Individuals who are obviously pregnant, nursing, younger individuals with known cardiovascular disease. That said, some might argue that, well, if it's an antioxidant, anti-inflammatory, and it may actually stimulate mood and it has no adverse impact on heart rate and blood pressure, why would that be a concern? Well, again, I can use the typical disclaimer of consult your physician or medical professional before using it, but I think obviously people need to be cautious and do their research up front, find out what they're using, make certain that the product they're using they trust, because we both know that sometimes even with CGMPs, et cetera, you want to make certain that what you're using and what you think is in that capsule is actually in that capsule. So, trusted sources, talking to some people who seem to know a bit about this particular ingredient or product, I think would be wise before starting to ingest things that they're not aware of. I think in general, if you look across the studies, and you're obviously aware of this and your listeners probably are too, we generally see with the Theacrine, especially when it's coupled with a moderate dose of caffeine, improvements in overall focus, improvements in subjective feelings of energy and vigor, vitality, things of that sort. And, outside of, a lot of times these products are marketed for sport performance, et cetera, and that's fine. But I think the reality is when you look at so many other products on the market that are targeting people that just want to feel a little bit better during the day, that market is quite large. And I think a lot of individuals, whether you're physically active or you're mentally active on the job, at home, whatever it may be, potentially could benefit from something like this. So it's something I think that in conjunction with a great diet, in conjunction with physical activity, regular exercise, hopefully people are doing those things because those are the most important things. Adding something like this to their regimen on a irregular basis or a few day a week basis might be something to consider.
Smart Drug Smarts.
So thank you so very much to Dr. Richard Bloomer for taking the time for that conversation. As he mentioned, he's got an upcoming study that should be published soon in the Journal of Caffeine Research, but still a lot of unknowns when it comes to theacrine. The mechanism of action not clear. Is it working on adenosine receptors the way that caffeine does? Probably the answer, as you would expect, is more research is needed, but definitely something of interest. Anything that can give that feeling of mental pep and vigor and alertness without making you feel like you need to get out and run around the block to get the physical charge out of your system. That's going to be of interest to a lot of people, myself included. A big hat tip to Michelle from the Smart Drug Smarts team, who got her hands on some theacrine, turned herself into an N=1 experiment, and says, for the record, that it tastes nasty. So we'll keep our eyes on theacrine. I feel like for right now, it kind of reminds me of a promising minor league baseball player. Hasn't made the big leagues yet, but somebody to keep your eye on. For right now, let's move on, as promised, to adult literacy and the brain in the Ruthless Listener Retention Gimmick.
Smart Drug Smarts, Ruthless Listener Retention Gimmick.
So as you probably know, the brain has certain areas devoted specifically to speech processing, things like Wernicke's area and Broca's area, among other uniquely human behaviors. We've been speaking for a very, very long time, but we've not been reading for a long time, and there are no brain areas that are specifically devoted to the skill of reading. There are plenty of illiterate human cultures that have been discovered. There has never been discovered a non-speaking human culture. You meet somebody that doesn't speak, it probably means they're just faking it and they don't want to talk to you. Reading is a different can of worms. We could certainly live to adulthood, and many people do, never learning to read. And learning to read is a pretty cognitively intense task. It's probably one of the toughest things that we throw at little kids, getting them to sound through the letters and figure out how to make words. You can kind of see the gears turning in their heads when that happens, and it's intuitively clear that there's probably a lot of changes in the brain taking place to support reading, but what those changes are have not been as fully explored as would be interesting, certainly. And since most kids learn to read at around the same age, it's also been tough to dissociate changes in the brain that might take place just as a result of aging and being at a certain developmental stage with those that are specifically tied to the skill of learning to read. One way to split this difference is to look at the brain changes in illiterate adults that are learning to read. And this proved to be a tough study for the researchers to conduct. It was tough for researchers to find a large enough sample size, a population of adult illiterates that were willing to come forward and say, yeah, I don't know how to read and I want to learn. So the research that you're about to hear about was conducted by the Max Planck Institute in Germany in combination with Indian scientists at the Center for Biomedical Research in Lucknow and the University of Hyderabad. India as a country has an illiteracy rate of 39%. Many people never have the opportunity to learn to read because they're limited by poverty and simply not having the access to education. The researchers identified 21 illiterate Hindi-speaking adult women. These were all women in their 30s, and they were taught to read the Devanagari script, which is the written form of Hindi, over the course of 6 months. And the results, maybe not surprisingly, were major. Scientific papers, which are not typically known for their extravagant language, they write in their conclusion that we have shown that only 6 months of learning to read leads to massive macroscopic functional reorganization processes in the mature human brain, which is about as close as you're going to see to getting a double exclamation point in a scientific paper. In addition to expected changes in the outer layers of the brain in the cerebral cortex, researchers saw major hemodynamic changes in the occipital lobe and subcortical areas in the midbrain and the thalamus. These are some of the older brain structures that are not by any means unique to humans but are shared among all mammals, so learning to read is having an effect at some really low-level areas, the extent of which was a surprise to the researchers. Hemodynamic, by the way, in case you're wondering, means changes in blood flow. Next time you're trying to hit on somebody at a bar and you want to tell them that they get your blood pumping, you can say, hey baby, you make me hemodynamic. That will probably only work out from you if she's also a Smart Drug Smarts listener, but don't let that stop you from trying. The level of connectivity increases between different brain regions were significantly related to the individual's decoding skill gains as they learned to read, says Michael Skida, the lead author of the study. We observed that the so-called colliculi superioris, part of the brainstem, and the pulvinar, which is located in the thalamus, adapt the timing of their activity patterns to those of the visual cortex. These deep structures in the thalamus and the brainstem help our visual cortex to filter important information from the flood of visual input, even before we consciously perceive it. We therefore believe that these brain systems increasingly fine-tune their communication as learners become more and more proficient in reading. This study also presented interesting questions into the possible origins of dyslexia. One possible cause of dyslexia has been attributed to dysfunctions in the thalamus, says Skide. Since we found out that only a few months of reading training can modify the thalamus fundamentally, we have to scrutinize this hypothesis. This would mean looking at the thalamus in brains of children that have not yet learned to read, following on to see which of those kids might develop dyslexia in the future, and learning whether there was an underlying problem with the thalamus to begin with, or whether there was some problem with the reading training that made the thalamus dysfunctional mid-process.
We like to think of ourselves as a digital speakeasy for brain hackers, but you can call us Smart Drug Smarts.
Okay, so you heard it. That was it for episode number 184. Thanks for hanging around until the end. If you enjoyed this episode, everything we talked about here can be found online at smartdrugsmarts.com/184. Last week in episode number 183, I spoke with Dr. Daniel Chow about his company Halo Neuroscience. Going to have a follow-up interview with his co-founder in a couple of weeks talking about implantable brain devices. Totally different topic, but equally interesting. And next week for 185, I'm not 100% sure what it's going to be, but very likely looking at the neural correlates of pleasure, basically how your brain has a good time, what your brain enjoys doing more than just about anything else. Popular guesses include sex and cocaine, but no, it's neither of those. But whether or not that winds up being the topic for episode number 185, I will see you back here next week, same time, same podcast, and with that unflagging, unwavering commitment to helping you fine-tune the performance of your own brain. Have a great week and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.